Okay, well, good evening, everyone, and thank you for joining this event, part of RSB's Biology Week. Welcome to our West Midlands branch charter lecture. So I'm very pleased to welcome friend and colleague, Professor Philip Young, to talk about spinal muscular atrophy. So a little about Phil and his work. Phil's worked on the diagnosis and pathophysiology of spinal muscular atrophy for over 25 years. While at Exeter, his research group focused on the cellular function of the survival motor neuron protein, particularly why motor neurons are specifically sensitive to SMN protein levels. And at the University of Warwick, Phil has focused on building a case for the introduction of UK newborn screening, a newborn screening program for SMA. So I hope that you find it very informative and enjoyable, and I know Phil is happy to take your questions at the end. Just before I pass over to Phil, just a little quick history of our charter lecture. So the charter lecture is to celebrate the Royal Charter that the then Society of Biology was granted in 2009. We've had many brilliant speakers in that time, many really good events, and I know that this one will also be excellent. So I'm going to stop showing my slides and hand over to you, Phil. Thank you very much.
So thank you all. So as Dan said, my name is Phil Young. I'm a professor at the School of Life Sciences at the University of Warwick, and I've worked on spinal muscular atrophy since 1996, so 25 years. So today I'm going to give you an update on SMA, and I'm going to talk about some of the novel therapies and the need for a UK screening program. So can we have the next slide, please? It is my Chris Witty impression. So I've split the talk into three different sections. The first section is on the background of SMA. So I'm going to go through the etiology and the pathophysiology. I'll then talk through the genetics. So I'll talk about the two different genes involved, the survival motor neuron genes, the SMN1 and SMN2. And then we'll talk about the genotype and phenotype relationship between these two genes and how they cause different forms of SMA. What I'll then do is talk about the really exciting developments, which is the SMA therapies. So up until recently, this was an incurable disease and an untreatable disease. But over the past sort of 4 or 5 years, two therapies have been introduced in the UK. So the first one we'll talk about is Spinraza, which is an antisense oligonucleotide therapy. I'll talk about what that means and how that works. And the second one is one of the most exciting advancements, I believe, in molecular biology and clinical medicine for a long time, which is the new gene therapy, which is called Zoltan Smart. So we'll talk about those two. And then we'll finish off the talk talking about screening programs, which is what I'm interested in. And we'll use the direct comparison between the US approach and the UK approach to highlight why we need a screening program in the UK. So can I have the next slide, please? So what is SMA? Spinal muscular atrophy. It's probably the most common genetic disorder that very few of you will have heard of. So etiology, SMA is the second most common genetic cause of infant death in the UK. Carrier frequency is roughly 1 in 50, and the incidence is about 1 in every 10,000 live births in the UK. So if we look at what causes the disease, it's caused by the loss of the anterior horn cells. These are the alpha motor neurons, which are the cells which link your spinal cord to your voluntary skeletal muscle. And when these die, what happens is you end up losing your motor end plates, and your skeletal muscle ends up dying through disuse atrophy. This predominantly impacts the skeletal muscle in the limbs, so the arms and legs. But in severe cases, it will impact the skeletal muscle, which is involved in your trunk, so your intercostal muscles. And when that happens, SMA becomes a life-limiting disorder. The next slide, please. So if we look at the actual classic forms of SMA, there are 3 forms of childhood SMA. So the first is type 1, which is the most severe form. The 3 forms are characterized by their age of onset and severity. So type 1 is the most severe form. Age of onset is roughly within the first 6 months of life. Type 1 patients never gain the ability to sit unaided, and with the natural cause of the disease, without any therapy, type 1 children will die within the first 2 years of life. Type 2 is an intermediate form. Onset of the intermediate form is roughly between the ages of 7 to 18 months of age post-birth, And basically type 2 patients can sit but never really gain the ability to stand unaided. And if untreated, type 2 patients will live beyond the age of 2, and their lifespan depends on various well-being and dietary sort of support and things like that. But nowadays, basically, we've understood how to look after type 2 patients. A type 2 patient in general would normally live until their 20s, 22, 23, that sort of age. The mild form of the disease is called type 3, so Kugelberg-Wieslander disease. Age of onset of type 3 is roughly after the age of 18 months. And type 3 patients, basically, type 3 is not a life-limiting disorder. It's characterized by generalized muscle weakness in the legs and the arms. But type 3 patients will be able to walk and to stand all the way through up into adulthood, at which point they may need a walking frame or a wheelchair. So next slide, please. So if we look at the genetics of the disease, what we know is that all 3 forms of the disease are caused by the same mutations. So it's an autosomal recessive disorder, which means one gene causes the disease. And that gene is the survival motor neuron gene, the SMN1 gene. So homologous loss of SMN1 causes all forms of SMA. And SMN1 is located on chromosome 5q13, which we know is one of the most unstable regions of the human genome. There are lots of different gene duplications here, deletions, gene conversions, recombination events, lots of things happen on this area of the genome. And one of the things we know has happened basically is that there has been a inverted duplication of the SMN gene. So SMN1 on this area, so if you look at the bottom, that's shown in blue in the red box, the SMN1 gene has been copied, inverted, and then duplicated, inserted back into the genome to form an identical, not near enough identical copy of the gene called SMN2. And we'll go through what each of those does in a second because they have big impacts on the disease. But we know that SMN1 is the ancestral gene, so every species and organism studied so far, right the way down to yeast, has a copy of the SMN gene. We know it's a housekeeping protein and housekeeping gene that has an important function. SMN2 is only found in higher order apes and humans. So we know that this inverted duplication of the SMN gene is a relatively recent evolutionary event. So can I have the next slide, please? So if we look at the 2 genes, both of them are near enough identical. They encode exactly the same full-length protein, exactly the same sequence, no functional difference between the protein that the 2 genes encode. However, if we look at the actual sequence, there are 5 single nucleotide polymorphisms between SMN1 and SMN2. And these have quite a large and severe impact on the actual processing of the gene at the RNA stage, if that makes sense. So again, SMN2 is an inverted duplication, and the important part is highlighted at the bottom in the boxes with the arrows. So there is a single nucleotide polymorphism in exon 7 of the gene. In SMN1, which is the telomeric copy of the gene, there is a C at position 6 of exon 7. If you compare that to SMN2, at the same position, nucleotide 6 of exon 7, this C has been converted to a T. So this is a polymorphism, it's not a mutation. So the change from a C to a T, even though it changes a codon, it's a silent change, which means the amino acid sequence encoded by these 2 genes is identical. However, this C to T transition has a huge impact in the SMN2 gene compared with the SMN1 gene. Can we go to the next slide, please? So what is the difference between the 2 genes? Effectively, it's that they produce different types of protein. So if you look at the SMN1 gene, it means that SMN1 encodes almost 100% full-length protein, and full-length SMN protein is the only functional form of this protein. So when you have the C in exon 7, you get correct processing of the gene and the RNA, which means you make full-length protein, which is 100% functional. If you compare that to SMN2, where the C has been converted to a T because of this polymorphism, what happens is that the SMN2 gene only expresses 10% full-length protein. 90% of the product in SMN2 is a alternatively spliced form or a truncated version of the protein that lacks one of the exons. This is called delta 7, it's non-functional, and it's degraded really quickly. So in general, SMN1 produces a really high level of functional protein. SMN2 produces a very low level of functional protein because of this C to T transition. Can I have the next slide, please? So let's go through some of the phenotypes and genotypes which we've seen, to be honest. So if you delete both SMN1 and SMN2, so both of them are completely removed from the genome, this is embryonic lethal. This is because SMN is a housekeeping protein. It performs an essential role in RNA production. It's involved in transcription, splicing, translation, and transportation stabilization of RNA. So if you delete both SMN1 and SMN2, this means that you don't actually make any SMN protein. What that means is that your cells can't make any RNA, which means they can't make any protein. And those cells which have no SMN1 or SMN2 become non-viable at the blastocyst stage during development. So this is a non-viable genotype. So there are no patients who have deletions in both SMN1 and SMN2. Next slide, please. If we look at mutations in SMN2, this has no clinical phenotype. So clinical phenotype in SMA is associated with SMN protein levels. So we know that SMN1 produces a really high level of protein. So if you've deleted SMN2, the level of protein expressed from SMN1 can compensate for the loss of SMN2. So 5% of the general population in the UK, when studied a few years ago, we identified mutations or complete deletions in the SMN2 gene. Again, this has no clinical phenotype, and you would not know you had a deleted SMN2. Next slide, please. In comparison, deletions in SMN1 cause SMA. The reason for this basically is if you delete the SMN1 gene, SMN2 is the only gene present expressing protein, and SMN2 produces very little full-length functional protein. This means basically SMN1 deletions and retention of SMN2 produces a significant reduction in SMN protein, and clinical symptoms are based on SMN protein levels. Next slide, please. And this can be expanded further. So as I said previously, the 5q13 genome is an incredibly unstable region of the human genome. And not only have we had an inverted duplication of SMN1 to produce SMN2, there are multiple copies of SMN2 which can be present on anyone's chromosomes. And what we find is that basically the copy number of SMN2 dictates the severity. So in a very simplified way, this is a slide which I present quite a lot. If you look at a type 1 patient, so the severe form of the disease, the genotype which causes this will be a complete deletion of SMN1 and one copy of SMN2. What this means is that the patient is producing a really low level of functional protein. If you compare that to a type 2 patient, again, no copy of SMN1 completely deleted and 2 copies of SMN2 per chromosome. So even though there is still a reduction in protein compared with a control individual or a non-SMA patient, what you see is you still have low levels of protein, but twice as much protein expressed as you would see in a type 1 patient because there are 2 copies of the SMN2 gene. If you then look at a type 3 patient, same thing. There are 3 copies in general of the SMN2 gene, which means even though there is clinical phenotype because there is a reduction in protein, because you have 3 times as much protein as expressed as what you would see in a type 1 patient, because there are 3 copies of the SMN2 gene, it means the severity is lower. So on the right-hand side is a nice summary of this. If you look at this is a control of protein level compared to an individual who has a copy of SMN1 and SMN2, which is the first control box. If you look at an SMN2 deleted individual, there is only a minimal reduction in SMN protein which is expressed because this individual still has a copy of the SMN1 gene. If you compare that to a type 1 patient where their SMN1 is deleted and there's only 1 copy of SMN2, there is a severe and significant reduction in SMN protein that's expressed. This is what causes the severe disease. This is the disease where the patients will probably die if not treated within the first 2 years of life. If you compare that to a type 3 patient, where there's 3 copies of SMN2, there is 3 times as much protein, which means even though there is disease, it is less severe than a type 1 patient because more protein is being expressed from the increased copy number. So take home message here, SMN1 mutations cause the disease and SMN2 copy number controls the severity. Can I have the next slide, please? But all SMA patients retain at least one copy of SMN2, because remember, deletions of SMN1 and SMN2 is embryonic lethal. So every patient has a copy of SMN2. And this means there is a viable therapeutic strategy, which led the NIH and FDA in around 2000 to highlight SMA as a disease that we could theoretically treat within 20 years. And thankfully, that's actually come true. So we know that all SMA patients retain a copy of SMN2, and we know that SMN2 is alternatively spliced because of a C to T single nucleotide polymorphism, which led to the question in the SMA field again in around 2000, which is if you can reduce the amount of delta 7, so the skipped version of SMN RNA, and increase the amount of full-length SMN protein expression from the SMN2 gene, do you treat the disease? In essence, a really simple question. If you treat an SMA type 1 patient and increase the amount of protein expressed from their one copy of the SMN2 threefold, do you convert type 1 to type 3? And the answer is yes. And the drug which has come out now, which does this, is Spinraza, which was approved in the UK in 2019. So next slide, please. What I'm going to do is I'm going to walk you through about 4 or 5 slides now, which explains RNA splicing, after which I can then tell you how Spinraza works. But I need to give you the basis of RNA splicing before I can explain how the drug works. So this slide here shows the central dogma of molecular biology. And that is that basically your genetic material in your genes is transcribed to form pre-messenger RNA. So pre-messenger RNA is effectively a single-stranded RNA copy of your DNA that contains exons, which are your coding regions, and introns, which are your non-coding regions. What happens then is that pre-messenger RNA has to be processed, and the processing is removal of the introns. And this is a process called RNA splicing. RNA splicing is the splicing out of intronic material to produce the messenger RNA, which contains the coding sequence. And it's this splicing event that goes wrong in SMN2. And the C to T transition has a huge impact on this RNA splicing process. So next slide, please. So to talk about RNA splicing, we need to talk about 2 different elements. We need to talk about cis elements and we need to talk about trans elements. And I'll go through these in the next 2 slides. So cis elements are the RNA sequences. These are the things that are present within the RNA. These are the binding sites, basically, which interact with trans elements. So for this talk, there are a multitude of different cis elements, but I'm going to focus on 3 specific ones. And they're all found within introns at this level. And these are the splice sites and the cis elements that define an intron. And remember, an intron is the part of the actual RNA that's going to be removed during intronic splicing. So the 3 we're going to talk about is the 5' splice site, which marks the start of an intron. That's the GU at the start of an intron. The second is the 3' splice site, which defines the end of an intron. This is made up of 2 different factors, the polypyrimidine tract and the AG site. And then the third is called the branch point or the branch site, which is an A, which is involved in intron removal. Next slide, please. So the trans elements, as I said, are proteins or ribonucleoproteins that interact with the C elements. And for RNA splicing at this level, there are 3 that I want you to get your head around. The first is called U1. And U1 binds and recognizes the 5' splice site. So U1 binding tells the splicing machinery that this is the end of an exon, this is the start of an intron, and we need to remove this part. The next part is U2AF, which is called U2 auxiliary factor, and this binds to the 3' splice site. Binding here allows the splicing machinery to define that this is the end of an the end of an intron and the start of an exon. And then the third one is U2, which binds to the branch point. And this is a really weak interaction, and this has to be held in place by U2AF. So for an intron to be recognized and then spliced out, you need all 3 of these factors to recognize the intron and then to bind to that intron. This complex, U1, U2, U2AF, is called complex A, which is the rate limiting step in RNA splicing. Next slide, please. Next slide, please. Sorry, thank you. So if you look at here, what happens if something goes wrong and you have an alternative splicing event? And all alternative splicing events is when the splicing machinery does not recognize an exon as being an exon. And the majority of time, this happens because there is a weak polypyrimidine tract, which has a very weak association or affinity to U2AF. So what happens in this instance here is U1 would bind to exon 6. So U1 is defining the start of the intron at the top diagram at the left-hand side. Before exon 7, you have a really weak polypyrimidine tract, which means U2AF cannot bind to it. That means the splicing machinery has not recognized that that is the start of an exon. What happens instead is U2AF will bind to the downstream polypyrimidine tract associated with the next exon, which is a strong polypyrimidine tract. So in this instance here, U1 binds and recognizes exon 6. U2 and U2AF do not bind to exon 7, they bind to exon 8. U1 will recognize U2, which means the exon associated with U1 will be spliced to the exon associated with U2, U2AF, which means in this instance, exon 6 will be spliced to exon 8, which means you get this delta 7 product where exon 7 has been removed. And this is what happens in SMN. So if we look at the polypyrimidine tracts that comes just before exon 7, so within it, intron 7 and just before exon 7 in the SMN2 RNAs, what we find is both SMN1 and SMN2 contain an incredibly weak polypyrimidine tract, which means ideally both of them could be alternatively spliced. Next slide, please. But what actually happens, the reason why SMN1 is correctly spliced and SMN2 is not correctly spliced, is this C to T transition within reason. So when there's a C present, which is SMN1, the C forms something called an exon splicing enhancer. So what happens here is, if you look at the diagram, U1 binds to the intron 6, so the start of the intron, to tell the splicing machinery that this is an intron that needs to be removed. You have a really weak polypyrimidine tract at the end of that intron, which means U2AF binds very transiently. However, because you have the C in exon 7, you form a splicing enhancer that recruits another splicing factor. This is a splicing factor called ASF-SF2, which has one function, and that function is to stabilize U2AF on a weak polypyrimidine tract. If you have a splicing enhancer, like you do in SMN1, ASF binds to it, it locks U2F in place, which locks U2 in place. And what that means is exon 7 is recognized as an exon in SMN1 RNA, which means 6 is correctly spliced to 7, and 7 is correctly spliced to 8. This means that in SMN1 processing, you always include exon 7, which means the gene encodes a full-length functional protein almost 100% of the time. Next slide, please. But in SMN2, at the DNA level, this C has been converted to a T. And at the RNA level, that means that the C has been converted to a U. What that means is that you lose the splicing enhancer. So in SMN2, instead of having an exon splicing enhancer in exon 7, you have something called an exon splicing silencer, an ESS. And this does not bind ASF-SF2, which means you do not get loading of U2AF and U2 onto the intron before exon 7, as you should do. What binds instead is one of the main inhibitory factors associated with alternatively splicing. This is a factor called HNRMPA1. This specifically binds to the U, which is now formed as part of the ESS. And what A1 does is it physically blocks exon 7. It blocks the polypyrimidine tract, it blocks the 3' splice site, and it blocks the start of the exon. So what it does is it masks exon 7, the start of exon 7. It means the splicing machinery can't recognize it. So that's one reason you get the alternative splicing event. There is a second reason, which is something specific to SMN2, which is there is a downstream intronic splicing silencer, which is highlighted in red and the arrow on the right-hand side. This is found downstream, and this ISS also binds HNRMPA1. And what this does is it blocks the end of exon 7. So these two splicing silencers, the ESS in exon 7 and the ISS in intron 7, both of which recruit HNRMPA1, means that you physically block both the start and the end of exon 7, which means the splicing machinery cannot recognize it as an exon. So what happens in most of the cases, around 90% of the RNA, U1 will bind to the end of exon 6, U2AF and U2 will bind to the start of exon 8, and because you've masked exon 7, 6 will be spliced at 8, which produces this delta 7 alternatively spliced RNA, which produces a protein, delta 7, that is really unstable and degraded really, really quickly. But again, it led to a therapeutic strategy or a research approach, which happened quite early in the 2000s, which is if we block either of these splicing silencers, if you find a way to block the ESS in exon 7 or the ISS in intron 7, can you increase full-length expression? And the answer is yes, or I wouldn't be presenting it. So can we have the next slide, please? That should be the RNA part. I promise, basically, we're going to move back into the interesting slides now. So the way that we tried to do this was the use of a really simple, but really efficient technique, which is antisense oligonucleotides, or ASOs. And these, as we move forward, are going to become really powerful therapeutic tools. So an antisense oligonucleotide, all it is a single small complementary RNA that binds and blocks a cis element. So in the box there, what you can see written in black, written in from start to end, so 5 to 3 prime, is an example cis element. So GGG, CCC, GGG, CCC, AAA, just a random sequence. RNA, when we're talking about the RNA which is being spliced, or anything like this, will be single-stranded. So an antisense oligonucleotide will be the opposite corresponding sequence. So read backwards, it would be UUU, GGG, CCC, GGG, CCC. This binds to the cis element in a complementary manner using Watson-Crick-based pairing, and it means that basically you physically block the C element because the trans elements that recognize cis elements can only recognize the single-stranded RNA. So the AOSs will block the cis elements and prevent, theoretically, trans elements from interacting. So I was part of a project which worked on this basically in the early 2000s, where we looked at all the different cis elements which have been identified to see whether or not any of the antisense oligonucleotides in a research setting can block the alternative splicing event in the SMN2 gene. Next slide, please. Because this is one of the most fundamentally important papers in SMA. This is the Ravinder Singh paper from 2006, I believe, yeah, 2006, which showed theoretically this works. And what they found is an antisense oligonucleotide that physically binds and blocks the ISS in intron 7. So shown there on the left-hand side with the arrow, this physically alters and corrects the splicing defects in SMN2. So if you look at the figure on the right-hand side, this is a figure from the paper. I apologize, it's quite blurry, but it does show what I want to show. If you can look at the two images there, you can see there, it's got the SMN2 splicing pattern. So you can see 2 bands in the gel. The top band is full length. So it's an RNA which contains exon 6, exon 7, and exon 8. So it's, and you can see in the SMN2 gene, that's quite weak. Comparatively, if you look at the bottom band, which is just exon 6 spliced to exon 8, so this is the alternatively spliced version, for SMN2, most of the RNA contains the delta 7 product. It lacks exon 7. If you treat these cells, bear in mind that this is from a patient fibroblast which is being used to treat this. If you treat these patient fibroblasts with Spinraza, what you see is a flipping of the ratio. So rather than most of the product being delta 7, what you now see on the left-hand side, which is the ISS N1, N3, 5 labeled with Spinraza, so this is just SMN2, RNA treated with Spinraza, you see most of the product now contains exon 7. So treating with Spinraza, which blocks the ISS, completely flips the splicing and corrects the correct splicing of the RNA. Next slide, please, because I want to really hammer home what that means. So this is a really nice figure. I like this one. On the left-hand side is the situation you have in a normal SMN2 RNA. You can see the ISS, which is folded around. That's the vertical sequence, which is underlined and in bold. That binds to the HNRMPA1. What that does is it physically blocks the 5' splice site, which prevents U1 binding. Remember, the 5' splice site is the cis element, U1 is the trans element. So if ISS is there and A1 binds, it means U1 can't bind to the start of intron 7. What that means is exon 7 can't be spliced to 8. What happens there is basically exon 6, which associates with U1, splices to exon 8, which associates with U2. What that means is the SMN2, the vast majority of the time, so 95% of the actual time, what happens is 7 is removed because your splicing machinery does not recognize it as an exon, and you end up with a delta 7 product. This protein is completely unstable, degrades very quickly, and is non-functional. If you add Spinraza, which is on the right-hand side, shown in red, it physically blocks the ISS. It means A1 can't bind, but it allows U1 to bind, which now means exon 7, the end of exon 7, is recognized as the end of an exon, which now means 7 is forced to splice to 8, which then allows 6 to be correctly spliced to 7. So the introduction of Spinraza allows 7 to be spliced to 8, 6 to be spliced to 7, and you get full-length RNA and resultant protein expressed from the SMN2 gene. Next slide, please. So brilliant. We showed it worked in cell lines. Follow-on papers from people like Arthur Burgess and some of them, the pioneers of SMA research in America, showed them that this worked in animal models. And then in 2017, this paper was released in the New England Journal of Medicine, which shows that Spinraza, that's also known as nusinersen, works in type 1 SMA patients. So as you can see here, this is a Kaplan-Meier graph. This is not from this paper. This is a combination of several different trials which have now been presented. And what you see here is a Kaplan-Meier graph. So this is individuals who are not given Spinraza, which is shown in the light gray line, versus patients who've been given Spinraza, which is shown in the blue line. And what this shows basically is everyone starts 100%, everyone is alive and on the trial. Every time there is an event, there is a vertical line. So the steeper the line, the more events that are happening. So as you can see, when you give when you don't treat patients, so the gray line shows the normal disease progression. And what happens here is it's monitoring the amount of events. And in this study, an event will be either a patient dying or a patient being put on permanent ventilation, i.e. then if they're taken off the ventilator, they will die. So it's near enough an end of life event. And the control event, the sham treatment tells you that basically the normal instance of a type 1 patient means that within a year, almost 70% of the patients will have either died or will end up basically on a permanent ventilator. So it shows the severe nature of the disease. If you treat with Spinraza, there is a slight difference. What you see here is only 40% of the patients end up either dead or requiring permanent ventilation, which shows Spinraza has had a significant and important impact and improvement on patient outcomes. Fantastic, it works, and it meant that basically this has now been approved in both the UK, the US, and most countries which are offering it around the world. Next slide, please. Sorry. So that's the summary of that slide. So part 2, Spinraza, just to explain what Spinraza is, it's an antisense oligonucleotide. It blocks the ISS and it allows the correct processing of exon 7. And more importantly, it works in patients and it's been approved for use, especially in the UK. Next slide, please. But, and this is where things get really exciting. I've got to keep an eye on the time because I do get excited and I go off at tangents. So let's make sure we keep to time. So there is an alternative therapy now, which has just been approved in the UK this year. And this, like I said, is one of the most exciting advances in medical science, I believe, in the past 20, 30 years. And this is the SMA Gene Therapy. So what is a gene therapy? First of all, a gene therapy is a modification of an individual's genes to treat a disorder. And remember, Spinraza is not a gene therapy. You are not changing SMN2 at the genetic level. What you are doing is modifying the processing of the RNA the gene encodes. What that means is the moment a patient stops taking with Spinraza, they start producing mainly delta 7 again, and symptoms can re-emerge. So what we would really want would be a permanent modification of the genes or the genetic makeup of a patient to prevent the need for continual Spinraza therapy or continual therapy with drugs. And there are two theoretical ways, one theoretical, one actual in practice now, where you can modify a gene using gene therapy. The first is CRISPR technology. We are years away from being able to do this, but theoretically, you can use CRISPR to convert SMN2 to SMN1. And the easiest way to do this would be to convert the T in exon 7 of SMN2 into a C, effectively allowing ASFSF2 to promote the correct splicing of the SMN2 gene. But we are years away from that, but work is ongoing on it. What is exciting is the gene delivery system, so the gene therapy. And this is Zoltan Smart, which has now been approved in the UK. So this is the use of a viral delivery system to introduce a new copy of SMN1 into patients. So can I have the next slide, please? So viral delivery systems, they've been around for a while, and there are a few issues you need to consider when you want to decide what you're going to use. The first is the viral delivery system needs to be non-pathogenic. So for example, you wouldn't use Ebola as a delivery system because you potentially could trigger hemorrhagic fever. So you're causing a problem rather than fixing a separate problem. So you need to choose a virus which doesn't cause disease. The second one is you need targeting. You need the virus to go to the right place. For SMA, you need a viral delivery system that's going to specifically target motor neurons. That's called tropism, and it's controlled by the proteins on the surface of the virus. The third problem, which is a big problem, is integration. So viruses, when they get into cells, a lot of them will integrate into the host genome. Doesn't sound like a problem, and more often than not, it won't be, because most of your genome will be non-coding regions. But if the virus integrates randomly into a gene or an exon, it can knock out expression of specific genes. And if it does that, it can cause a secondary complication, which is a real problem. So when they looked at this and they developed this Zoltan Smart, they went and used adeno-associated virus serotype 9. The reason they use this is AAV causes mild to no associated disease. It's a DNA virus, which means it's very easy to manipulate, and it's easy to modify it so that it specifically targets motor neurons. Can I have the next slide, please? So this is a very simplistic slide of what Zoltan Smart actually is. So if you look at the top schematic there, that's the AAV9 in a very simplistic way, showing you what the genome encodes. So there are two ITRs at the end and the start and the end of the genome. These are involved in genomic integration when the AAV9 enters an infected cell. You then have the rep loci, which encodes all the proteins that are needed to drive viral DNA replication. You then have the cap region that contains all the genes that encode protein that are involved in viral coat and capsid production. And both of those are needed to replicate the viral genome and then encapsulate it in a capsid to make new viral particles. What you do in Zoltan Smart or any gene therapy is you remove the most important proteins. So you knock out rep and cap and simply replace it with the SMN1 gene, which means this virus no longer can replicate or produce new virus. The second thing you do is you mutate the ITRs, which means this virus, the Zoltan Smart, cannot integrate within the host genome. So there's no secondary complications of integration, and you can't make more virus, which means you're not going to make or cause any downstream problems. So if we go to the next slide, sorry. What you would then do is you take this new modified genome and you would package it within a capsid, an AAV9 capsid, which contains viral coat proteins that specifically target the virus to your motor neurons. And this produces the SC-AAV9 viral vector that is used to deliver SMN into cells. What you then do, you purify this and you inject it directly into the patient's spinal cord, where it can unpackage, and then your delivered SMN gene will be transcribed, spliced, and then translated to form functional full-length protein. Next slide, please. We are getting to near the end. I apologize if I'm going too late. And this paper basically came out back to back with the Spinraza Zoltan Smart therapy. This is the single dose gene replacement therapy for SMA. New England Journal of Medicine 2017, this was published. This basically shows that if you introduce this virus into patients' spinal cords, there's 15 patients here, there's now hundreds of patients who've been treated with Zoltan Smart, but in the initial paper, what it shows is that their muscle function improves, there is an improvement in motor milestone reaching. So this is a measurement using chop intend, which is a way of measuring whether patients are meeting their motor milestones. So patients who, and these are all type 1 patients who are given Zoltan Smart, all improve and reach a point where you would class it as normal motor function within 12 weeks, sorry. And what we find there, if we go to the next stage, is that this basically is well tolerated in patients. It's FDA approved, and now it's been NICE and NHS approved for use in the UK in 2019. Can I have the next slide, please? Which is the graph. Oops. Right, so this is the, if you look, this is the same graph I showed previously, which is the Kaplan-Meier graph. The eagle-eyed among you will have noticed the green line at the top. So as we've said here, this graph shows, if you look at control individual, roughly around 70 patients have either progressed, died, or required permanent ventilation if they aren't treated. Compare that to around 40% of patients who have been given Spinraza who have progressed, died, or required ventilation 12 months after the initial treatment with Spinraza. If you compare that with Zoltan Smart, which is the gene therapy, which is the horizontal line at the top, there are no recorded patients who have died or required ventilation who've been given Zoltan Smart, telling you that this is one of the most efficient and effective therapies that I've certainly been exposed to or seen during my time basically as a researcher over the past 25 years. It is a phenomenal therapy. Spinraza is also a phenomenal therapy. However, there are problems with both of them. The first is Spinraza is very expensive. Spinraza is the second most expensive drug ever made. It costs £150,000 per injection, and patients will need 3 injections or 2 or 3 injections for the rest of their lives. Incredibly expensive. Zoltan Smart is the most expensive drug ever made. It costs £1.79 million per injection, but it is a one-off therapy. So we've gone from having no therapies to having 2 therapies for SMA, both of which are available in the NHS, but both of which are incredibly expensive. Can I have the next slide, please? We are getting to the end, I promise. So summary, Zoltan Smart is an AAV9 delivery system. It delivers SMN1 gene into the motor neurons of patients, and it restores normal full-length protein expression. And the results are spectacular. This is the first major gene therapy which has been introduced for a neuromuscular disorder, so it is phenomenal. Have about 4 slides left, so I promise I'll move on. So if we go to the next slide, which are 2 final questions. The first one, when should we treat patients? And I'm going to walk you through the American system and then compare it to the UK system. And then I have 1 slide on whether or not we have truly cured SMA. Next slide, please. So in the US, following the approval of Spinraza and Zoltan Smart, they also introduced a newborn screening program. So in the US, currently in most of the states, there is an SMA newborn screening program, and that was introduced in around 2018, 2019. So in the US, most children, if not all children, certainly in most states, are tested within 2 days of birth using a heel prick test. And if a child is diagnosed as having SMA, they are given either Spinraza or Zoltan Smart within the first 2 weeks of life. Really important, because there is, if you look at the mouse work and you look at some of the clinical history work from patients, we know there is a 16-week therapeutic window from birth to when symptoms emerge. And if you treat patients within that window, you maximize the impact. And the reason for that is because the cells you're trying to treat. Motor neurons do not replicate, and symptoms emerge because neurons are dying. So if you are treating after symptoms emerge, it means you are stabilizing surviving motor neurons rather than saving all the patient's motor neurons. So remember, the earlier you can treat, if you can treat patients before they're symptomatic, you have a better chance of surviving and saving more of their neurons. Next slide, please. Because if you compare that to the UK, that's not what happens. In the UK, just a reminder, 1 in 50 UK population is a carrier statistically, but there is no UK screening program for SMA. In the UK, SMA is diagnosed when patients become symptomatic, and most patients have no family history of this disease. So in the UK, even though we are using these incredibly expensive drugs, we are administering them after symptoms have emerged, which is outside the 16-week therapeutic window, which means theoretically, we are reducing the chance of having a true and sustained and long-term impact because we are stabilizing surviving neurons rather than saving the ones that we could have done if we'd have treated pre-symptomatically. Next slide, please. Very quickly, there is only like 2 or 3 slides left. I know I've said that 4 times. So this is what I'm involved in now with Felicity Boardman, who's a phenomenal professor at Warwick Medical School. We are trying to get a UK screen involved. We're trying to get one introduced. So in 2018, Felicity was involved and I was involved in generating evidence for this. We submitted a request to the National Screening Committee to get a newborn screen for SMA introduced. Next slide. Unfortunately, though, it was rejected. This is just a very clear timeline just to show where we are at the moment. So in 2018, NICE initially rejected Spinraza. The reason they rejected it was because of its cost. NICE, which is our regulatory body in the UK, which approves drugs and recommends drugs for use in clinical practice, NICE would normally only recommend a drug if it costs between £20,000 to £30,000 per quality adjusted life year. Spinraza costs about £400,000 per quality adjusted life year. So NICE said that Spinraza was too expensive, so they rejected it. What happened then was, this was just after we submitted our request to the National Screening Committee, and based on the rejection of Spinraza, the NSC rejected newborn screening for SMA. They rejected it because there was no therapy, so they saw it was immoral and unethical to diagnose the disease in the general population. About a month after NSC rejected rejected the screening program, NICE did a U-turn and approved Spinraza and said that any UK SMA child who is symptomatic could now be treated with the actual drug. And then in 2021, May 2021, NICE have now subsequently approved Zoltan Smart, and I think 2 children in the UK have now been treated with it. Again, once they're symptomatic. So now what we're trying to do is trying to get a NICE, an NSC introduced screening program so we can maximize therapy and outcomes for patients similar to what we're seeing in America. But the submission for that won't actually happen until 2022. Next slide, please. And this is just what we're doing now. We're involved in the National Screening Alliance, myself, Felicity Boardman. We are going towards a NSC application in 2022, 2023. So last slide, I promise, and I'll leave you with a final question, which is basically, all this is fantastic. I have gone from basically in 1996, when I joined the SMA field, we'd only just identified the gene. Within 20 years, we had a potential therapy. And then with 25 years, we have 2 drugs that are actually now in clinical practice. The level of expansion is amazing. However, there is a very clear question. Have we cured SMA, or have we simply changed the disease? So for a long time, SMA was thought to be a neuromuscular disorder. So if you fix the neuromuscular disorder, you would then cure the disease. However, if you look at the SMA mice, when they've been on Spinraza or Zoltan Smart for a long period of time, they start developing secondary complications. They develop problems with their liver, with their kidneys, with their heart, with their pancreas, which could mean the fact that basically SMA, because of the nature of what SMN actually does, which is RNA processing, and the fact that it's a housekeeping protein, SMA could be a systemic disorder that just happens to present in motor neurons. So the neuronal pathology, the fact that it presents in motor neurons, may be because these cells are incredibly sensitive to SMN levels, and the fact that when they die, they don't replicate. So we could have problems in all cells in the body, but the fact that basically hepatocytes and cardiomyocytes and these sort of cells, they may be dying in SMA patients at a higher rate, but because these cells replicate, you don't see the clinical pathology. And because we know Zoltan Smart and Spinraza are both neuronal specific therapies, there is a potential and a worry within the field that all we've simply done is cure the neuromuscular disorder and converted SMA to a different type of disorder that may cause clinical issues down the line. However, that's what we're focusing on. We have a window where we can now see whether this is the case and then move forward and improve the therapies. That's it, that's my last slide. I'd just like to thank all the people who've helped me over the time, especially Felicity Boardman, who I collaborate with at Warwick. Then the students, so Raksha, Oliver, Chloe, Elsita and Farah, all of whom have been involved in SMA research projects here at Warwick. And then I have Chris Lawson, who was my postdoc supervisor in Missouri, and then Glenn Morris, who was my PhD supervisor. Thank you, and I'm happy to take questions.